Be the girl of my dreams. I wait for you. In our pajamas, that faded bananas, monkey see, monkey do. Would you be the girl of my dreams? Don't make a monkey of me. Space correct, free activity. Be the girl of my dreams. Oh, not our place, it's your branch from of all of my mates, I know you'd be prime. You'd be my prime, you'd be the girl of my dreams. I will be free. In a perfect job, you see, wouldn't you be the girl of my dreams? Don't make me monkey of me. Sorry, be the girl of my dreams. When you let me rub a scarlet on the roof, I told all the people that you're simply fantastic. Be the girl of my dreams. I will be free. In her job, that day, would you be the girl of my dreams? Don't make me give me. Sorry, activity for your captivity. Be the girl of my dreams.
This is TWIS. This is Circumcising in Science, episode number 497, recorded on Thursday, February 18, 2015. TWIS takes Manhattan. I'm Dr. Kiki, everyone, and this is This Week in Science, and today we are going to fill your heads with an implant, drunk birds, and circumcision. But first.
Disclaimer. The following program contains explicit educational language, graphic fiction, scientific fact, and brief full frontal disregard for intentionally supernaturally derived notion of how stuff works. It is intended for mature and irreverent listeners of all ages. Subject matter covered may be objectionable to people whose ideologies are rooted in the Middle Ages. All that is required for listeners to enjoy the show is that they listen. If after listening you find yourself challenging people to know before listening, the desired effect has occurred. If you find after listening yourself pondering relative strangeness of reality, the desired effect has occurred. If after listening you find yourself deciding nothing is better for your brain than giving it a view outside of your skull, the desired effect has occurred. And if after listening you find yourself wanting more of what your brain likes, more is available at This Week in Science, coming up next.
I've got the kind of mind I can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Good science to Kirsten and Blair.
Good science to you, Justin and Blair, and everybody in the house. Yeah!
Yeah, for the first time in This Week in Science history, we've mic'd up my usual live audience.
That's right.
Yeah.
The live.
Yeah, they're always here. They're always with us. This time we actually got them mic'd up, so you can hear them. You can hear them enjoying the show along with us.
Yeah, the bleachers in your living room.
Mm-hmm.
I know. Those ones, yeah. We are here in New York City, in Manhattan. For the first time, the three of us are actually broadcasting together in the same location. We're almost to our 500th episode. And we've never been in the same
We're bad
place.
coordinators.
Yeah.
About this sort of thing.
We had to travel thousands of
And
miles.
actually, we're
to get
all
this
within
done.
an hour of each other.
Yeah.
But it took us going to a different coast to etch out the time to be actually in the same room together.
Yeah, so I'd like to thank Erez and everyone at Horse Trade and the Under St. Mark's Theater for giving this chance to be live broadcasting. Thank you so much. But it's time for science, because that's what we're here for, right?
Right?
You got it.
Yeah.
Yeah, I got it. You got it. Okay, on today's show, We have tons of science news. I have some stories about finding resistance, some questions, and a fancy new spinal implant. What do you have, Justin?
I have bad iPhone relationships, where dogs come from, and a potentially correlative study about circumcision.
You're mincing your words there. Blair, what did you bring?
Well, you know I love alliteration, so I brought buzzed birds, fertile frogs, and fizzling fish.
Awesome. Let's get on with this sizzling science production. Okay, everyone. My first story of the night is about dun, 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 neural interfaces for your spinal cord. What? So people with paralysis, we have no way currently to get severed portions of the spinal cord talking to each other again to reverse paralysis. And there are implants that people have been working on for years, but the materials have been substandard, and so if you put the implant in, it usually causes inflammation, it causes scarring, and it ends up damaging the spinal column much more than maybe just leaving it alone and letting it heal naturally, or dealing with it in some other way. So these French researchers, French and Swiss researchers, have been working on creating soft neural implants. So it's taken a team of biologists, physiologists, materials scientists, of computer scientists to develop algorithms and engineers to be able to get the materials all put together. And they've created the first truly soft, flexible, tissue-like implant that can actually be implanted under the protective coating of the spinal cord, the dura mater, and slipped in there right on top of the spinal cord, where it can deliver electrical shocks, it can deliver chemicals also, and it doesn't cause scarring. They tested it on rats, in which they had severed the neural column, and in severing the column, the spinal cord, the rats were paralyzed. These rats could not walk. They stuck these little implants on the spinal cords of the mice. They gave them electrical stimulation and neurotransmitters that would get the nerves talking to themselves, talking to each other again, and all that combined with physical therapy, rehabilitation therapy. After about 2 months, the rats were walking, and there was no scarring, and the implants were being completely, the body was not rejecting them.
That's freaking amazing.
Yeah,
so
that's
for,
so
for we've been doing the show for
Almost
more
500
than
episodes.
more than a decade.
Yeah.
And we've covered maybe 50 different stories of one shape or another that are trying to figure out how to circumvent this, how to go around, create a loop circuit outside of the spinal cord, to go directly to the legs, to power an exoskeleton to get people to be able to walk again. But if we have a potential to actually create a cure, that's an amazing, huge breakthrough.
Yeah, so they're going to be starting clinical trials and trying to actually move from rats into humans, because once again we have cured something in a rodent species to be something we're really good at. So let's get the clinical.
They're expendable, that's why.
Yeah.
We use a lot.
So we're going to get the clinical trials going in humans, but it's not just paralysis that this could be good for, because it can be implanted within the neural, basically the brain and the spinal cord. We could be looking at treatments for Parkinson's, Alzheimer's, paralysis. It's almost limitless what we could do with an implant like this.
Yeah, and this required so little therapy and training on the part of the rat that they were working with, which with so many other paralysis fixes that they've been trying, it requires so much time trying to get them to relearn how to use their brain or some sort of big switch, but they were using the mechanics already in place for the most part, so that was super cool.
Yep, letting the body heal itself.
Yeah.
Making it easy for the body. I like it. The second story that I thought was really, really exciting that I wanted to lead off the show with this week had to do with resistance, antibacterial resistance. So we have people here in the audience, you've been to the doctor, they've given you antibiotics, or you've taken maybe a child to the doctor and they've said, well, your baby doesn't necessarily need antibiotics. Let's try not to give your baby antibiotics right now. Let's wait for something a little bit more serious. The reason they're doing that is because antibiotic resistance is a huge problem. All of the antibiotics we have now are currently, there are bacteria resistant to them. So
they've stopped, the bacteria have evolved the resistance. Or we've just killed off all the bacteria that had the weakness. And now only the strong are surviving.
Only the strong
so,
survive. Yeah.
And there is this fear that we could go out another 20 years and not have antibiotics. Like what we have just won't work anymore because it's run its course.
Yeah, so vancomycin is the most powerful of our antibiotics, and basically what bacteria had to do was steal the protein from the bacteria that produce vancomycin to protect themselves. So what we do is we mine bacteria for their own antibiotics, and those are the antibiotics that we use on ourselves. So back to the bacteria we use, that we mine, basically they put out these compounds, and they have to have something that allows them to protect themselves, give themselves a little force field from whatever that compound is. And so there are proteins involved in that usually. And so those genes, those proteins were co-opted by other bacteria, and so now that resistance is spreading everywhere. But this cool new study is so exciting. Okay, so bacteria that we use for antibiotics, we've gotten them from dirt. And we took all the bacteria that we could get from dirt and cultured them in labs. And then we're like, oh, you've got this antibiotic, oh, you've got this antibiotic, this works great, that works great, we're going to use these, fantastic. But we ran out of bacteria from the dirt that we can culture in the labs. Like, there are 99% of the bacteria on Earth we cannot culture in labs. They do not live in a dish in the lab. For whatever reason, we do not give them what they need to survive, and we can't grow them in the lab.
Can't's a big word.
Well, this is the new thing. So, so these researchers came up with this special box with a lot of holes in it where they tricked bacteria. They stuck it in the dirt, and these holes were really tiny. They were just big enough for one bacterial cell. And so they got bacterial cells isolated in these little corrals, basically. It was like a little farming operation for bacteria. And they let them grow in their little corrals until they created little bacterial colonies, and they took those bacterial colonies and they mushed them all up, and they said, what did you make? And they say, put them in a blender, and they're like, what stuff did you make? What did you make? And do you kill other bacteria, and what do you kill? And they did 10,000 of these analyses. Took 10,000 different bacterial cultures, and they ended up finding one, a compound that they're calling teixobactin, and teixobactin inhibits Staphylococcus aureus. It inhibits MRSA. It inhibits, let me see if I can find all the other ones. It inhibits pneumonia causing bacteria. It inhibits and kills all these bacteria, and they couldn't find any bacteria, specifically MRSA and Staphylococcus aureus, that had any kind of resistance to this teixobactin. So this is a new antibiotic. It was well tolerated by mice that they infected with contagious, infectious bacteria.
So again, we've cured another disease that affects them.
Yes. Yeah. And then they took human cells and they put them in a dish, and the human cells tolerated the teixobactin as well. So human cells lived and tolerated it. And so the next step, again, clinical trials, but this is something that's really promising. And the big excitement is that we've gotten it. They found that this bacteria they got it from is from, there are two classes of bacteria, gram-negative and gram-positive bacteria. Gram-positive bacteria have a cell wall. Gram-negative bacteria have this fancy protective membrane outside of their cell wall. So they use, their cell wall is structured differently. So far, all the antibiotics we've been using are from gram-positive bacteria. This is from a gram-negative bacteria. So the problem with the proteins getting transferred is probably not going to be a problem because it's from a completely different type of bacteria, which is a really good thing. And secondly, the resistance is going to be different because instead of going after proteins, it goes after lipids, the fat molecules that are used to create the cell walls. So it stops bacteria from being able to build their cell walls again. And so this could be a really big advance in antibiotics. Better than anything we've done. But anyway, I don't want to get anyone's hopes up.
Right. Yeah.
What
So
made-
I shouldn't go like lick one of the park benches in Central Park. Like
I
I'll
want
be
my
fine.
Taxo back in!
what major pharmaceutical company research came up?
I don't know, but it was from a pharmaceutical company and a university collaboration. I don't remember the answer. I don't know the answer.
They're gonna corner the market.
They're going to corner the market on that for sure. Right. That's a different story. Hey, Justin, what you got?
iPhone separation anxiety. So this is.
I've got my.
Does anybody here out in the audience? Does anybody of you use a cell phone or a smartphone device of some sort? It's pretty much
None of them.
unanimous.
None of them at all.
Anybody.
No.
So, yeah, this is research at the University of Missouri has found that cell phone separation can have serious psychological and physiological effects on the user, including poor performance on cognitive tests. The researchers say these findings suggest that iPhone users should avoid parting with their phones during daily situations that involve some degree of attention, require you to be paying attention to something, such as taking tests, sitting in conferences or meetings, completing important work assignments. This is awesome. So this is a doctor's note for any situation where somebody's telling you not to bring a cell phone along. It's
Sorry,
damaging.
science.
Science has said you have to
I
keep
know
it with you.
in high schools, they collect cell phones at the door to keep for tests and other stuff. They shouldn't.
Not anymore. Now, well, then all the
No.
students who are listening to this can bring this in.
Maybe it hasn't set in yet with students. Maybe it's only for adults
Oh,
who
no.
have,
No, I think it's-
responsibilities and maybe children or loved ones that are trying to
Higher
hold
stress
them.
levels.
We were kind of talking about this earlier. If you've ever gone to work and forgot to bring your lunch with you, have you ever gone to some place where you didn't need to spend money and forgot your wallet and just didn't bother going back and getting it? And how many times have you been like you're about to be late for work, you're halfway there, you realize you didn't bring your cell phone, you go back home, right? You go back home. You have to have this thing with you. So yeah, the findings suggest iPhone separation can negatively impact performance on mental tasks, says Russell Clayton at the Missouri University School of Journalism, lead author of this study. Additionally, the results from our study suggest that iPhones are capable of becoming an extension of ourselves, such that when separated, we experience lessening of the self, a negative psychological state. Right? So this isn't just
It's your identity. You've lost your identity.
Take my hand. Don't take my iPhone.
Well, think about it. Okay, so it is part of your brain now. It is part of your brain. If you don't know something, you don't concentrate really hard to try to remember. You just Google it. You just go to the Wikipedia. You just try to find that answer right away. When you no longer can do that, you already feel like part of your brain is missing. Part of your access to information literally is gone.
Yeah, well, we were talking about phone numbers earlier, right? So maybe 10, 15 years ago, you had phone numbers memorized in your brain, and now you don't memorize a phone number. It's in your phone. So then if you don't have your phone, what are you going to do? What's wrong? And you need to call someone important.
So also think of it, even if it's not important. Even if it's totally not important, we're so used to the ability to communicate to anybody in our network instantaneously that if you lose it, I sort of imagine it as losing the ability to speak. We're so used to talking to people who are around us.
Losing
right?
the ability to use an arm.
Yeah.
Well, no, I think it's bigger than that. I think it's a loss of ability that we take for granted now, which speech, before you had a phone, you just talked to somebody,
So
But-
now the studies that need to be done need to specify exactly how big of an effect it is. Is it an arm or is it your speech?
Right. So here it is. So
Those are
the researchers asked iPhone users to sit at a computer cubicle in the psychology lab. They had the participants first they had them do a word search puzzle where they had like a scramble. You had to find the words in there with their phone. They got to have their phone with them. And then they said they were testing. Technically, they said they were testing a wireless blood pressure monitor. So they had their arm in the thing. And then halfway through the test, they told them that the iPhone might somehow be disrupting the Bluetoothy technology of the wireless cuff. And so they took it and they put it across the room. They didn't like take it away from them and put it in another room where they couldn't see it. They took it and set it like across the room on a table.
People are just stealing glances at their phone.
Right?
Yeah. No, I think that makes it
What's going on over
worse.
there?
You're like, I wonder if it's vibrating over there.
So-
Does it miss me?
Are there any messages? Do I have any notifications?
No, listen to this. It wasn't just anxiety, general anxiety. Their heart rates went up, blood pressure rose. They had a physical reaction to being separated from this piece of technology. So, a couple of things. What does this look like? So I got a couple of things here. I was trying to think of what this looks like. Do we define this as codependence? Is this like a relationship with the phone? Is this like an unhealthy love affair where we're like so obsessed with the other that we just can't be away from them? Because it kind of trumps most of our actual relationships in that way, I think.
Yeah.
Right?
Sometimes, yeah.
And then,
You mean like when you're at dinner with someone you really care about and you're on your phone the whole time? More important than the person that you chose to spend time with?
right. And then the other part of it too
child
is
you chose to have.
there's also that sort of scary side of it too, where we all have abilities to locate our missing phones now. So in the case that our significant other, being our iPhone, is not near us, we can stalk it at any time. We can track its every motion.
All right. This is This Week in Science if you are just tuning in. And you know what time it is? It's time for Blair's Animal Corner.
You guys choreographed that earlier, or?
Oh, yeah, we spent hours.
Yeah,
Hours.
nice. I'm flattered. So let's jump right into a story that I've actually been saving for a couple weeks. It came out right around New Year's Eve, and it's about alcohol. And I'm sure we all had a few drinks on New Year's Eve, and we all kind of have different theories of how it changes us and how it affects us.
Makes
Our
me more
various-
handsome.
Well, to some people.
To myself.
But yeah,
That's
to
how it
yourself.
counts.
You put your own beer goggles on
Yeah.
and then you look in the mirror, right? So it affects our cognitive abilities, though, in a lot of different ways. We might start to slur our speech. We might have trouble balancing, and we might start to stumble back and forth a little bit. Also might make us want to do things that we might not normally want to do. Well, scientists have not been really sure how it affects our brain, actually, what it's doing to our brain, what it's doing to the various areas of our brain that have these effects on us. So it's hard to pinpoint it in humans. It's really hard to perform studies on humans on how alcohol affects the brain. So they did it on some birds, and Kiki's favorite birds, zebra finches. Yes, they sound exactly like that. And they gave some zebra finches some harmless white grape juice, and they gave some other zebra finches a mixture of juice and ethanol. And they watched what happened. They watched what happened to their bird song. They watched what happened to their movements. And they found that it changes the bird's amplitude and entropy in their songs. They weren't able to sing as loudly, and they couldn't keep their songs on a normal, stable structure. So they kind of likened it to slurred speech. And so they kind of got some clues on which part of the brain is getting disrupted when it's changing our speech. But they saw, this is what's really weird, no change in motor abilities. None.
So even though a drunk human might stumble and not be able to walk in a straight line, the birds can fly totally fine.
So we're still not sure why that happens. But so they watched the birds. The birds were able to fly totally normally. They didn't have anything that relates back to what we consider human drunken face, like being able to have normal wing structure. They weren't drooped in their wings. They weren't closing their eyes more. They weren't acting sluggish.
You said human drunken face?
Yes, that is literally in this study. Human drunken face. Yes,
My
that's
new
one of the
favorite.
reasons I love science. Okay, terms like that. But so they didn't have any of these things. All they had was their slurred speech in their song. So they were able to kind of get a clue into what area of the brain is being affected. But yeah, still totally a mystery why it makes us clumsy.
Yeah, that's fascinating. I know you have an increase in GABA, or is it a decrease in GABA? There's something related to
You get an increase, I know,
GABA
from
and-
doing yoga.
Yeah.
So
lots of inhibition
It
in
seems
the
like that
brain
would be-
generally.
Yeah, inhibition's a whole nother one. We didn't even
Yeah.
even talk about that really. It was talking more about
Well, alcohol acts as an inhibitory effect on the brain, so it's like adding up increases a whole bunch of the inhibitory neurotransmitters and decreases the stimulatory ones, and so that's potentially, if it's a broad brain effect in humans and not in birds, the question as to why that happens.
Exactly right. So if it was a broad kind of overall effect of the neurons firing slower in any animal, then we would have seen that in the birds. But for some reason in the birds it wasn't doing that.
Dude, I want to be a bird.
Yeah, just slur your speech.
No,
but
then
be
you
totally on it. Otherwise,
wait,
Totally on
doesn't
it.
it sound
Yeah.
like then alcohol just wouldn't work? It sounds
Except
like a
to make
horrible.
you, just except to make you sound
I guess they didn't care about that. So.
really awful.
Yeah.
Oh my goodness.
Yeah. So that's the bird song. But let's talk
Drunk
about
birds. It's not a British story. It's
Yeah.
really a bird story.
It's a bird story. Let's talk about some vertebrate sex today.
Not invertebrate.
No, I have no
Vertebrate.
invertebrates today.
Vertebrate sex.
Only vertebrates.
That
so,
means related to you guys. Us guys.
I mean, any fish out there? No. So not really.
Well,
We came from fish.
we came from
Yeah,
fish.
totally.
You had gills in the womb. Come on.
Yeah. So fish, ghost knifefish. First of all, coolest fish name ever. Ghost knifefish, right? If I was going to be a fish, that'd be a pretty good one. They use electricity in their courtship, which is pretty cool. We know about other animals, eels, obviously, using electroreception and using electricity for other things. We also know about sharks, of course, using electroreception, other fish in there, a lot of the time for prey capture. But this is for copulation, for attracting a mate. So that's totally new.
I could zap you.
Yeah. Hey.
What? Hey.
I'm real electric over here. Yeah. Turn up the electricity. So that's literally what they
I'm
do.
sure this happens at some kink castles, but
Yeah. Okay. So anyway, the fish. The
Anywho.
fish.
Do go on, Kiki. Tell
Yeah,
me
should
more.
we pause? Let's table the fish, and you can explain what that
I got
means.
more interested in a different story. Now, what was
No,
this you were
fish.
saying?
we were talking about fish.
Okay, so all right, we'll get back to that side table. All right, so the ghost knifefish, they have a specialized electric organ in their tail that makes an electric organ discharge, an EOD, and they have receivers all over their skins to receive that information, the EODs, and they interpret their signals. And what they found was that the females, they recognize EODs from each other, but the males have a completely different frequency from the female. The females do 600 to 800 hertz, and the males do 800 to 1100 hertz. So it's a totally different arena if you're making electric impulses as a male or a female ghost knifefish. So I'm just going to keep saying that name over and over, ghost knifefish. So actually a pretty good Halloween costume. I have to remember that. All right, so anyway.
You're gonna have like 100.
I know. So the ghost knifefish, they have a variety of hertz that they emit depending on the individual, and each individual has a different kind of signature of their EOD. And what they found was that the females really preferred the higher frequency. The higher the frequency, the better the male. So there could be a whole different number of reasons for that. It's possible that it takes more energy to make a higher frequency impulse, which means they're more fit if they can use more energy on electricity. There's a whole bunch of different theories you could go with for that. But what they found that was even more interesting was that they emit the most EODs during rainstorms, which is their breeding season. So again, that's something that if their weather changes, that could totally mess up their breeding season if the rainy season changes. But even more interesting, when they took these fish out of the wild and experimented with different social groups in the lab, they adjusted how high their frequency was. If there were more males around, if they were around a lot of other males, they would kind of beef it up and try harder. So-
and we've seen this actually, something very similar when studying human population, where they were testing the amount of juice men could produce, and when men watched pornography that involved
Other men.
not just other men, but a man and a woman, like the female videos didn't really do anything, but the male-female videos, it triggered some sort of competition in them that they produced more juice after than previously.
There you go. That's intra-specific competition,
Yeah.
right?
Exactly.
So even the fish do it.
And I guess for this particular species of fish, we could say love hurts.
No, you can't say that one.
Bum. Okay, last story
I
for
have
the
one
animal
more.
corner. One more
I
story.
have one more. It's about frog breeding. So it's less about the mechanics and more about what happens afterwards. A critically endangered type of frog called the white-spotted bush frog, found in South India. They've been found to breed in a way that no other frog or toad has ever been seen breeding. They do it inside bamboo. So what's crazy about this is that these frogs were thought extinct. And in 2003, they were found again. So this is definitely, there's almost none of these guys around. We thought they were totally gone. There's a few left. But part of the reason we had trouble finding them is that they're hiding out inside bamboo shoots.
Not just inside growing areas of bamboo, but in the bamboo.
Inside the bamboo. So they
How get in there?
they're really tiny. They're about 25 millimeters. That's 2.5 centimeters. That's like an inch. So they're very small frogs, and they squeeze in through the small slits. The males do. Then they call out for females. The females are brought in. They come in to the calls, and then the females lay a clutch of eggs inside the bamboo, and then they leave, and the males stay, and they take care of the eggs. And this is where it gets really interesting. Frogs, everybody knows they make tadpoles, and then they turn into froglets, and then they turn into frogs. Well, these guys come out of eggs as froglets because it's dry in the bamboo, so they can't swim. So they come out already with arms and legs, and while they're hanging out as eggs and when they're froglets before they're large enough to leave, the male hangs out, and then he will leave for a few hours to feed, but then he'll come right back again. He'll hang out there until they're old enough to come out. Now, this is the 41st type of reproductive mode found in frogs and toads. They find every possible which way to do it, basically.
Nice.
And
No
by do it, I
frogs.
mean raise their babies. So this is interesting because these guys are critically endangered. We want to save them. We found out what is key for these guys to survive, the bamboo. And the bamboo are really commonly harvested for paper and pulp.
But you said in your prediction show, if you could find a reason for pandas to be alive in this world,
Yeah.
pandas, conservation, we're going to be conserving bamboo forests for these little frogs, which is
Already.
going to save the pandas. You're dressing up in a panda suit, Blair.
I'm 1 week into the new year, and it's already happened.
You did it to yourself.
I'm very upset. All
Long
right.
live the pandas.
Well, that happened. Now, I have to tell you, my favorite thing about this story is a quote from one of the team members in the research, Dr. David Bickford, who's from the National University of Singapore's Department of Biological Sciences. This is one of my favorite science quotes ever. It's 2014, and we are still making discoveries like these. Natural history is sexy, always was and always will be. No matter what the molecular and genomic revolutions have accomplished for us in the biological sciences, nature is still the ultimate source for everything we do in biology.
Nice.
He's a pretty cool guy.
Seems like he would be.
Yeah.
Yeah. And on that note, that is the end of the first half of This Week in Science. We will be back after a very short break. Stay tuned for more. TWIS has merchandise that you might enjoy. We have a Zazzle store online. If you go to our website, twis.org, and look for the Zazzle link, you'll find t-shirts and hats and stamps and tote bags and other things that are fun with the TWIS logo emblazoned all over it. So if stuff is your thing and you would like to support us that way, I urge you to go to twis.org and look for our Zazzle link. Additionally, we are supported by listeners like you. Your donations support everything that we do. So we have a couple of ways to allow you to support us. We have a PayPal link, or lots of PayPal links, all over our website. Once again, twis.org. Go there, look for the PayPal links, click on them, and we give you lots of options to be able to support us financially. Second way is through Patreon. Patreon's kind of like Kickstarter, but for media producers. So you can support us on an ongoing basis, show to show, and you choose your level of support. And in return, you get fun gifts. And as I'm sitting here at the Under St. Mark's Theater, I've got some of these fun gifts out in front of me, patches and stickers and pins and other things that we send you in return for your support. So your money becomes the source for everything that we do, and it will help us grow and do more science for you. We also understand that donations are sometimes very difficult. Financial support comes hard to some people, so if that is something that is difficult for you, we accept support in all sorts of ways, like telling your friends and your family, your loved ones. Use your social networks for science. Tell them about TWIS. Tell them we're fun. Tell them they might like it. You know, send them a link. That kind of thing. We thank you for your support. We really could not do what we do without you. And now, a song from Glenn Raphael.
Thank you. This song is based on the many worlds interpretation of quantum mechanics.
Not what you were expecting, huh?
Which, as you know, is a theory in which every possible universe exists, and that means that if you make a choice and you could have done something else, some other version of you makes that other choice in some other universe. So this is either a love song about physics or a physics song about love, and it's called Quantum Entanglement. I'll say. Every little decision splits us in two. There must be a million worlds we've been through. Before I go, there's something that you ought to know. Most of me are happy with you. I mull this over at home in the bath. Every choice we make creates a new path. Smarter men than any of me tell us there must be worlds we agree. That's not a metaphor, they did the math. This quantum entanglement, spooky action at a distance, whole world's a possibility. There is only one I see. You know I love you, but also I don't. I will be there forever, but also I won't. I'm sure most versions of me love almost every version of you. There are worlds we're here for, everyone we're through. This quantum entanglement, spooky action at a distance, whole world's a possibility. You're the only one I see. In the world beyond Niels Bohr, what are decisions for? Do they change anything? What will our future bring? We have a moment with luck, it might last. All our actions, reactions are now in the past. This much is true, as anything any of me ever knew. Most of me are happy with you. Thank you.
And we're back with more This Week in Science.
Oh yeah, more science for everybody. Justin,
I've got a canine mystery.
canine tooth.
No. This is a study that did some genetic research into the American dog, the North American, South American dogs, ancient dogs. These aren't the modern ones, the ones that we're seeing today. These are ancient dog burials. They found 84 individual dogs to study in this, and what's really piqued my interest about this is they're dating the Americana dog to about 10,000 years.
We've had dogs for 10,000 years.
Well, yeah, that alone isn't that special. But in the Americas, the furthest back they can figure that there were dogs here is 10,000 years. Now, dogs and people together, that can go back maybe as far as 16,000. People in the Americas, it's about 14,000. That's about the, well, I mean, and probably longer than that. They found late 13,000 years ago South American sites. So there's a puzzle. There's some missing time here.
Yeah.
Right? There's a missing time of about 4,000 years when we were here with no dogs. And
Or we were here with dogs, but not leaving traces of it. Or,
possible. But this is, I mean, everything from occasionally eating a dog to burial of dogs with a little bit of ritual to it. So very cared for members of the tribe and occasional snack. But the thing that's sort of just odd about this to me is, and it's not that they re-domesticated dogs here. It's not like there were two separate dog domestication events. Some of the dogs do have a lineage to the American wolf, but it's sparse. Most of it is not related.
See, and that's very different from the narrative that we've always heard, that there was the gray wolves here and that they domesticated them.
Yeah.
Yeah, that's crazy.
Yeah, most of them, dog domestication is thought to have originated, and it's a wide margin. They're between 11 to 16,000 years ago in Europe. So we have Europe, which is over, there somewhere, with the Americas, which is over here, which we had to go off the map and wrap back around to get to in the first place. But maybe the dogs, maybe it's like when the owner moves and forgets to bring the dog miraculously finds the owner 4,000 years later. But it made me start thinking, like, maybe this is because whenever we find one of these puzzles, and it could be that this gets pushed back and they find more evidence and they find a way to secure the dog coming across the Bering Strait with us. But if they don't, what it implies is basically a trade route. That there may have been a lot more than a single crossing, a single event, and that there may have been a lot more connection from the Americas back into Asia.
And there has been some evidence, like, just of peoples at different times, archaeologically, that seem to suggest there are a couple of crossings, the evidence is starting to kind of push more in that multiple crossing or even trade route kind of.
Yeah, I mean, that's
interesting.
So this, what this little study on 84 buried dog remains that they found implies, possibly, is a rewriting of the Americas' origin story.
It's a pretty big implication from just some
Dogs.
dogs. Yeah, that's
dogs.
wild.
Dead dogs, big implications. I'd like to take this opportunity. I've got a story I want to open up to comment from the audience. We have a microphone that Glenn was using, so if anyone wants to comment on this particular story, I'd love to hear what you think. about synthetic biology and what is going to be happening in the near future, where things are going to be going.
Can I start off the?
Can I just tell the story?
Well, I can. I just want to put my 2 cents in before the story. I'm for it.
Good to be on record.
It sounds good already.
It's like the AI and the robots.
All for it.
Okay, there we go. So future robots, AI, and synthetic organisms. Justin was for it. He's always on your side.
Let it be recorded for all time.
For all future robot masters, if you look back into the archive and hear this recording, I was for you from the beginning.
That's right. That's right. Okay, so genes, right? We have the 4 letters of our DNA, ATCG, adenine, thymine, cytosine, guanine. These things that, when put together, create us. They help us create proteins and all the things that build us. But lately, it has become an alphabet, and it's an alphabet that we're learning not just to read, but to write with also. And there are companies, like a company called Cambrian Genomics in San Francisco, led by Austin Heinz, where they are printing DNA on demand. And so it's not necessarily DNA that is sequences that are in existence in bacterial species already. It could be DNA sequences that somebody is making up and trying to create a whole new organism or a whole new function for existing organisms. And the 2 questions that I think are really important here are, should we be using synthetic biology to solve our man-made problems? Things like soil erosion. Should we be engineering bacteria to have some kind of function that makes trees grow roots a little bit deeper and a little bit stronger, that holds onto the soil a little bit better so that it doesn't erode away as quickly? Should we also make the language of DNA publicly available? Should it be free for all? Should it be open source? What kind of regulation should be on it? And with companies like Austin Heinz's Cambrian Genomics, they have started a startup laboratory called Creature Creator, in which they are inviting people to come up with small business ideas to create creatures using synthetic biology. And they're using Kickstarter to try and fund these things. Austin Heinz was part of the group that made glowing plants, I think was the thing that they made. They make glow-in-the-dark plants. Fluorescing plants was the first thing that they made, and there was a big Kickstarter to make that happen, and you could buy the plants, get them by supporting the company on Kickstarter. And so they're very successful at using Kickstarter to get funds to create these companies, and they have, I'll give you a couple examples of the companies that are currently in the works. One is Petomics. They're making a probiotic for cats and dogs that makes their poop smell like bananas. I don't like the smell of bananas personally.
Yeah, but if
So
you had
I don't know
a
if it's any
choice,
better.
but Kiki, if you had a choice, if you've got 2 choices,
Bananas, I don't like how you smell. Poop, I don't like how you smell either.
How will you know when to clean the litter box then?
It smells like bananas.
But what if I also bought bananas?
You are flat out of luck. And then the second company is called Sweet Peach. And this is a fun one. This one is hoping to take samples of users' vaginal microorganisms to send back personalized probiotics to promote vaginal health. Forget the douche of the past.
Oh.
yeah, no, this is awesome. This is a major step forward.
I
I'm alone on this? I thought that the audience would be like, yeah, no, but this is the proper usage.
just pictured you in their campaign, Kiki. Forget that douche of the past.
Oh no. Oh no. Anyway, these are the kind of simple and fun-loving ideas for startups, but his idea really is he wants thousands of these kinds of startups, and he wants to try and make the tools available to people to do this whenever and however they want. And so, and he also doesn't want a lot of governmental regulation. He wants regulation by the companies, for the companies. He says it's in the company's best interest. So, how's that going to work out when there's lots of companies DIYing whatever organisms they want and printing DNA on demand? I don't know. It's just kind of a lob out there. If anyone in the audience has anything to say, throw it out there. There's a spotlight. Oh.
Hello, this is Byron Lee.
Hi,
Eighth
Byron.
Dominion. Pleasure to see you.
Yeah.
Really, what could go wrong? All of our other biological experiences have just been great. But am I correct that we would have no genetic resistance to any of these new things?
Or they may have no genetic resistance to us. I mean, we don't know. It's a complete unknown. The thing I would compare this to is sort of the chemical revolution of the 40s and 50s, where we were producing all sorts of new chemical combinations, new materials, and they were going out into the environment. And we had no idea what the repercussions were going to be. Some of them were very negative. A lot of them, I'd say the vast majority of them, were extremely positive. So it does mean you're putting yourself in the way, in harm's way, by allowing companies to do this. But the potential reward, I think, ultimately would outweigh the risk.
Yeah, I wonder. From the biology standpoint. Blair, you can probably talk to this a little bit more, but I mean, there are numerous examples of us trying to control things, use one species to control the spread of another species, and having it go horribly awry.
Yeah, introduced species are pretty much always a mistake when we're using the actual species. When we introduced bullfrogs, when we introduced any number of things that we've introduced to other places, specifically to eat pests and stuff like that, a lot of the time they turn around and eat exactly what we don't want them to eat. So it's one of those things where you have to really understand from absolutely every single angle whatever you're trying to replace does. And so many animals, we have so many or other biological kind of elements of an ecosystem, we don't always have the whole picture of their function. And so it's really hard to kind of predict that until they're gone. But I can see lots of times we've lost what we consider keystone species, where we take out an animal or a plant species that just totally tears apart a habitat without it. It totally collapses the food web or something else going on in that area. And if we could find a way to replace it until things stabilize and then we can reintroduce, we can find a way to do that, that could be really beneficial. The question is, how do you control it once you let it go?
So I'm for it.
Right.
Blair, you've carefully balanced an argument with yourself and against yourself. Kiki?
I like to do that.
I like to sit on the fence as well. I'm a good fence sitter. No, I think I have a real question about this personally. I think there should be a big conversation between the government and also, I mean, and I'm not talking just the United States government, I'm talking like the global governments, and DIYers and people who do garage science and people who are doing startups and creating new companies. And there should be a conversation about this because there is the potential for it to go really wrong. But I think if everybody is having a conversation and trying to work together, for the betterment, then, it's going to be great. And as Austin Heights thinks, it's going to be an awesome 100 years.
And
I'm sure
really,
it sounds exactly like that.
Right?
much like
It'll be
the
his
chemical revolution, I think it will take a little bit of effing things up for the regulation to come in.
Which is unfortunate, but
Well, yeah, but I mean, this is also a learning process. I mean, this is how we educate our government, is by allowing companies to do terrible things, come up with terrible ideas, and then going back and regulating after the fact. It's worked pretty well.
so I think what we need to do is we need to build some sort of biodome, if you will.
We'll put them in
False
a
sense of security.
Put them in a box. and we'll keep 'em in
Know
the box.
that you're wrecking things. Just know that you're wrecking things, possibly wrecking things, and then be vigilant about it. But yeah, I think it's going to be tough to regulate something that the regulators don't have any grasp of. I mean, we don't have scientists running this country. We don't have people who are going to be able to positively, put proper regulation in there. It's always going to be too lax, or it's going to be too severe, and then they're going to take, potentially some, what would you call it, supernatural ideological reason for being against a science blanketly, and prohibiting its pursuit. And so I think it's better. We're better served by leaving the government out of it, because history has shown us in something
Potentially.
like the stem cell research,
Mm-hmm.
when the government gets involved, it's for the worse.
It's not good for the research at all. I wanted to bring this up because I think it's something that everybody should be aware of in terms of the research that is ongoing. And we are at a stage in synthetic biology where we are going to probably this year create the first print on demand where it's not DNA from an existing organism that's had DNA spliced into it, like a single gene giving it one new function, but a complete DNA, a complete chromosomal makeup printed from scratch, made up by scientists. It'll be an organism that survives in the lab. It's going to happen. Probably this year, if not this year, then next year it's going to happen. And that's going to be something that's going to be more and more common. We'll be able to take printers, DNA printers to Mars, and print bacteria to terraform Mars if we want to. I mean that's one possibility. And
Mars manifest destiny.
that's right.
Talk
And
about
in-
science fiction.
and
This
in a
is
way-
really science
and in a
fiction.
way-
But it's starting now,
Coming.
and so people should be thinking about it and
Yeah.
thinking about how they want how do you want your future to go? What products do you want on the shelves? Do you want that fancy douche?
Yes. Who wouldn't want that to be available?
You want pets? Poop to smell like bananas?
But the other thing is too,
Why
like
not
whenever we talk
strawberry?
about this, it sounds like this is a completely brand new boundary that science is breaking. That oh my gosh, we're not going to manipulate an organism. We're going to create our own.
It's creating our
It's
own.
creating our own. We don't know what it'll do, but that happens every time when we do manipulate. When that happens, this is what antibiotics are. I mean, we could have been afraid that antibiotics were going to wipe out all bacteria, our healthy gut bacteria, or everything else. I mean, we could have censored all of science for the fear of the result.
Yeah.
But we, whenever we go through one of these scary phases where we're doing something new, somebody comes out there and makes it sound very frightening. Edison electrocuted Topsy the elephant to show that alternating current was gonna destroy all of our lives. Yeah,
And many
but
cats,
it was
too.
kind of a
Many cats.
mean elephant. Well, okay, he shouldn't have been in captivity. But still, the point is, it's not that scary. When we get past that unknown factor and the reality sets in, it's never been that terrifying, except for one physics concept that splitting an atom kind of did get the whole world on edge for a little while.
little bit,
With the
but-
exception maybe of that one.
Yeah. Well, why don't we talk about something else that's kind of scary to some people? Your next story.
Oh.
Okay. So this is research published the Journal of Royal Society of Medicine, suggesting that circumcised boys are more likely than intact boys to develop autism spectrum disorder (ASD) before the age of 10. Risk, they say, is particularly high for infantile autism that sets in before the age of 5. So this research was carried out in Denmark among a cohort of children born between 1994 and 2003. During the study, over 340,000 boys were followed up on the age up to age 9. And 1994 to 2013, and almost 5,000 cases of ASD were diagnosed out of that population. As it's going to be pointed out, I'm sure. We don't know what percentage here, unfortunately, of boys were circumcised. It's pretty low rates. And
Didn't you look it up, Kiki? Isn't it 2%?
We
Less
don't know
than
what the-
2% of the
Yeah,
population
small
in Denmark
population.
are
Small
circumcised
population.
currently.
Yeah, they showed that the study showed that regardless of cultural background, circumcised boys may run a greater risk of developing ASD. They also made the unexpected observation, this is kind of a side here, of an increased risk of hyperactivity disorder among circumcised boys in non-Muslim families. Don't know.
We don't really
That's
understand
really
that
co-
result.
Correlative, maybe, to the
Uh-huh.
culture and the genetic makeup.
Changing your tune there from earlier, Justin,
yeah,
are you?
I am. You got a little feedback when I was bringing this story that not everybody was on board with me. So it says their investigation was prompted by the combination of recent animal findings linking a single painful injury to lifelong deficits in stress response, and a study showing a strong positive correlation between countries neonatal male circumcision rate and its prevalence of ASD in boys. So the sort of the premise here,
Correlation is the premise.
it is a correlation, but the premise here
Correlation.
does make sense to me. So what they're saying is because it's an extremely painful traumatic event
When
that's
they're
happening
1 day to 3 days old.
when they're right at the beginning of development, when development is
Right
where
after they've been smooshed out that little tiny birth canal.
which is how nature's been doing it for a really long time.
Yeah, and they're fine with it.
And they're fine with it.
They're fine with that.
Without any painkillers.
So okay, so
Just saying.
I'm gonna share a little bit with the audience here and say I no longer have my foreskin, and I can't get it back. I've asked several doctors if there's a way to reattach. Apparently, it was discarded. They threw it away. They didn't even preserve this thing. They didn't even give
I
me an
don't think
option
you
or a
would fit in
choice.
it anymore, Justin.
Yeah. You'd be surprised. So the thing is, if you can imagine, this is your first or second day on planet Earth. Maybe it happens within a week, and it's like, okay, there's light, there's temperatures now. Temperature varies. That's something new. And then they don't use full anesthetics on infants. And then this surgery takes place. And you can say, oh, there may have been some local anesthetic applied. But the healing process after this continues. This is an extremely sensory overload, one of the greatest collections of nerves in the human body. And you've just done a traumatic, painful surgery to it. And for somebody who's just new to the plan, I can see how that could create a reluctance to be involved in social matters going forward. Developmentally, as a protective reaction for the brain to say, you know what? The less we interact with these people out here, the better. Let's just keep it all inside.
So there's
Nothing
so
good will
many
come of
things.
it.
Point. Okay. Yeah. Blair and I both. Point taken.
Yeah.
We accept your argument.
Respectfully disagree.
Respectfully disagree. So, animal studies. These are taken from mouse studies in which they showed that these early painful experiences led to changes in stress response. Changes in stress response are not equal to autism spectrum disorder. That's a complete different.
That's apples and oranges for sure.
Yeah, completely different. So we're looking at two different traits, and so they've taken and then looking at, oh, well, these countries seem to have more autism, and so hence correlation and kind of baseless assumption. Let's jump into this study and get another correlation. And blah, 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 blah.
And so I think that they probably could have had access to a huge set of data for America.
Yeah, why don't we look at a bigger database?
Yeah.
Yeah.
Because 2% of 300,000 is not a lot, and
we
then
don't really know what the makeup of that less than 2%
Yeah.
is. So who are the people in that small population of people who choose to have their children circumcised? We do not know that.
So if we really want to look at this, maybe we should look at a country where the majority have circumcision. So then it crosses all different types of variables that you could be looking at, so you can actually do proper science.
Right, and this is part of the problem. And I
Proper
agree.
correlation.
I kind of agree with both of you, because here's the trend that I've noticed in watching this subject over the however long we've been doing the show, is that whatever country is doing a report on the proposed health benefits or the adverse effects of circumcision, it completely correlates to the nation's position or what percentage of the population is doing this. So if there's a lot of circumcision,
Have you gone back retrospectively and taken all those data points?
well, the one that
Yeah.
the World Health Organization trying to claim that circumcision reduced chances of getting HIV
in
by
Africa.
50%. It was a totally ridiculous study.
It was by 50%,
It was not. It
although
wasn't.
that is
It was
statistics where the change was from something like 1.5 to 3%
Yeah,
of the population that they're
wasn't.
looking at. So they're looking at a very small percentage of the population.
And the percentage of population that had the interactions with the doctors where the other ones didn't, because one had a medical treatment and the other hadn't. So there was all sorts of stuff that gets left out. But it seems like every time I hear one of these stories, it completely fits with what the social norm is of that country. So no real good science being done on circumcision.
I think we can leave it at that.
Yeah, I would agree with that.
I think we'll just leave it at that
For
statement.
sure.
We can agree to disagree, but we agree on that.
Yeah.
Okay, so I have a couple of headline news stories as we head into the end of the show. So a couple of this week in the world's first time that scientists have observed DNA shuttling between regular cells to cancerous cells. They saw mitochondrial DNA being squeezed through nanotubes from a healthy cell to a cancerous tumor cell in which the mitochondrial DNA had been removed. Thus, the cancer cells renewed their ability to grow, and the cancer started growing again. So there is DNA in humans, in cancer, in humans, this mitochondrial DNA is shuttling between cells. And they think it might be a lot more widespread, so they're going to look into that more. And additionally, world's first time that scientists have gotten mussels to grow in a dish and to contract and respond to electrical stimulation and to chemical compounds in a way that will allow them to study mussels in a dish so that they won't have to be on an animal or a person. And these mussel cells were stem cell they were stem cell progenitors, so myogenic progenitor cells. So ideally what could happen is that we could take stem cells from somebody suffering from some kind of muscle disorder, grow their cells in a dish, and they would be a perfect model of what's happening in the body of that person.
That sounds peachy.
And then we live forever, right?
And then we live forever.
Yeah. Then beginning and.
Yeah. Did you have a headline story?
Yeah. An amazing thing happened at the California Academy of Sciences back in San Francisco. A shark had viable eggs, and babies hatched out of this shark egg 4 years after that female shark had ever seen a male shark. 4 years. She stored sperm for 4 years.
That
First
would
they-
make for a very long and involved Maury Povich.
Yeah.
episode.
Yeah. Let me go back. Think about every male shark
We've
I've
got to
ever
talk
been
to
with.
everybody you've talked to
Yeah.
over 4 years ago to figure out who the daddy is.
Yeah. So they thought it was parthenogenesis at first, where the female just asexually reproduces. She makes clones, but they tested the genes and they weren't clones of her. So it turned out it was from a boyfriend
not
4 years ago. So this is the longest ever for sharks recorded. Yeah.
Of course, I didn't load up my Patreon stuff. Once again, sorry to log in very quickly.
Hey, well, you didn't, this I don't have in front of me, but there is again renewed talk that and going to be looked into here this year and next. Potential that the tachyon, this theoretical particle that moves faster than the speed of light and therefore backwards in time, may actually be a neutrino. We may already have this within our grasp. It may already exist, and the implications of this are pretty amazing. One of the things you could conceive of is if you find a way to emit these muons, these neutrinos, to go backwards in time, you may be able to create a neutrino detector where we have one. This doesn't work like we need it to yet, and receive messages from the future because it's moving backwards in time, right? So what you would have to do, though, you wouldn't actually have to perfect today the art of sending this neutrino anywhere. You just have to have the detector there. All you need is the detector, and because then someone else, somewhere down later on, can create this.
The sender,
Yeah.
the transmitter.
Yeah. So we could
Like
just,
it.
Perfect.
and then we could figure out. We could ask, like, so how do you make one of those senders? And then it would just show up, and we could right? We could just do like time capsules where we're asking the future to solve our problems, and it would just keep showing up, and it's like, oh, it's another recipe for how to cure another disease, or
And then at what point am I my own grandmother? When does that happen? It's eventually.
I don't think that's how that works yet. No, but you wouldn't have to like play sports ball anymore, because you could just ask who won.
Who's gonna win?
Right.
For
Yeah.
like, and the teams could save a lot of money, right? They wouldn't
Well,
have to
it
pay
is.
players
It is
anymore,
2015.
and we
It
could
is the
still
year of Back to
bet
the
as long
Future.
as nobody
So
knew the outcome,
yeah,
right?
Exactly.
perfect.
It is that time, everyone. We have come to the end of our show, and I'd like to give shout-outs to those of you who are sponsoring us on Patreon. Thank you to Kevin Donald, Wesley Ballard, Paul D. Disney, Sam Winzier, John Ratnaswamy, Rudy Garcia, Steve DeBell, Greg Guthman, Stephen B., Jason Dozier, Matthew Litwin, Eric Knapp, Andreas Akestad, Jason Roberts, Patrick Cohn, Shannon Tara Ginsburg, Byron Lee, E.O., Bob Calder, Jared Lysette, Ulysses Adkins, Jake Jones, Edvardas Rimkus, Nicholas Lai, Brian Hedrick, Kathy Lester, Chris Cross, don't you wanna? Makes you wanna. Sarah Chavis, Stephen Sorowiak, Layla, Paul Stanton, Marshall Clark, Charlene Davidson Henry, Don Kamarachka, Randy Mazuka, Ed Dyer, Tony Steele, Lord Trentonian, Moody, Larry Garcia, Daryl Lambert, Brian Condren, Richard Bowersox, Kevin Parachan, Dave Major, Brian Rossman, James Dobson, Steve Goodwin, Kurt Larson, Stefan, Michael George, Craig Landon, Russell Jensen, Mountain Sloth, Jim Drapeau, Darren Luke, Tara Payne, Alex Wilson, John H. Maloney, Jason Olds, James, Paul West, Lauren Lang, Ed Bennett, Thomas Mikinen, Alec Doty, Illumalama, Joe Wheeler, Dougal Campbell, TMRO, Miko Pakala, Craig Porter, Adam Mishkan, Laren Luthen, Marjorie, Scott Lajewski, Tyler Harrison, Ben Rothid, Colombo, Ahmed, Gary Swinsberg. Thank you. Thank you for your support on Patreon. If you are interested in supporting us, you can find information at patreon.com/thisweekinscience. Also remember that you can also help us out simply by telling your friends about TWIS. On next week's show, once again, we're going to be on the air live, broadcasting at 8 p.m. Pacific time on Wednesday. This was a special show on Thursday on twis.org/live, where you can watch and join our chat room. Don't worry if you can't make it, though. You can find our past episodes at youtube.com/thisweekinscience or at twis.org.
Thank you for enjoying the show. TWIS is also available as a podcast. Just Google This Week in Science in your iTunes directory, or if you have a mobile device, you can look for TWIS for Droid in the Android marketplace, or This Week in Science in the Apple marketplace, or the iPhone podcast directory thingy.
For more information on anything you've heard here today, show notes will be available on our website, www.twis.org. That's TWIS.org. There you can also make comments and start conversations with the hosts and other listeners.
Or you can contact us directly. Email Kirsten at kirsten@thisweekinscience.com, Justin at twistminion@gmail.com, or Blair at blairbaz@twis.org. Just put TWIS, TWIS, somewhere in the subject line, or your email will be spam filtered into oblivion. You can also hit us up on the Twitter, where we are at TWIS Science, at Dr. Kiki, at Jackson Flynn, at Blair's Menagerie. We love your feedback. If there's a topic you'd like us to cover or address, suggestion for an interview, please let us know.
We'll be back here next week, and we hope you'll join us again for some more great science news.
And if you've learned anything from today's show, remember,
It's all in your head.
This Week in Science. This Week in Science.
thank you, New York.
This Week in Science.
Thank you, everyone, for
Thanks,
coming.
guys.
This Week in Science. Is the
Thank
end
you
of
to the
world.
theater, Perez, Will, Nick.
Got
Thank
my
you.
banner unfurled. It says the scientist is in. I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. This week's science is coming your way. So everybody listen to what I say. I use the
I didn't bring
method for all that it's worth. And I'll broadcast my
But,
opinion
I'll find it.
all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got one disclaimer, and it shouldn't be news. That what I say may not represent your views, but I've done the calculations and I've got a plan. If you listen to the science, you may just better understand that we're not trying to threaten your philosophy. We're just trying to save the world from jeopardy. Jeopardy. And This Week in Science is coming your way.